This video is about the process of protein synthesis. We've learned about DNA structure, how it replicates. We've also learned about genes and how genetics works. But now the question is, how do we get from DNA, all these A's and T's and C's and G's, to you, or to any living creature? What is the process of taking those instructions that are in the DNA and using them to actually make something? The first thing you need to understand about this is the something that is being made here in the process is proteins, because it's all about the proteins. So remember that proteins make up a lot of you. They make up your hair and your muscles and your skin and your pigments and all kinds of things. They also make up all of your enzymes, and your enzymes perform chemical reactions or facilitate chemical reactions. So your enzymes are what builds all of the other things that make up you. So the DNA has the instructions to make the enzyme, which then makes the other things that aren't protein. So by making proteins, essentially the DNA can make or control the production of all the things that make up you. So we're going to start with a story. It's a little metaphor. You don't need to take notes on this first part of the story. You can just listen. All right, well, my cat has come to join me. I don't know if you can hear purring, but if you do, that's what that is. So the story. We are at a candy factory. And at the candy factory, there is a boss. But the boss likes to stay in his office. Perhaps he's a sort of more rotund individual, and he is not actually able to get through the door. He's too big to get through the door of his office, and that's okay. He likes it in his office, so he just stays in there. But he relays messages to his messenger, and he tells the messenger, go take my instructions out to the candy factory floor, because he can't leave, so he gets the messenger to go out and give his instructions. So out at the candy factory floor, the messenger arrives and he gives the instructions to the workers. And he tells the workers what ingredients to bring over to make the candy. So this worker maybe brings over the sugar, and this one brings over some coloring. So each worker brings over each ingredient according to the boss's instructions. And finally, all the ingredients are put together to make the candy. Yay. So that story is, in fact, a metaphor for how the process of protein synthesis works. So now, instead of the boss, we have DNA. And instead of the boss's office, we have the nucleus of the cell. So the DNA stays inside the nucleus of the cell. It cannot get out of the nucleus. But it sends out a messenger called messenger RNA, good name for it. This can be abbreviated mRNA. And that takes the instructions from the DNA, like a piece of the DNA's instructions, a copy of it, out of the nucleus to the ribosomes, because the ribosomes, if you recall from organelles, are what actually makes the proteins. So out in the ribosome, the mRNA with that message from the DNA comes in, and then the workers now are called transfer RNAs, also called abbreviated tRNA. And so the tRNA bring over the amino acids, because amino acids are the building blocks of proteins. They're the monomers of proteins, so those are the ingredients, according to the instructions on the mRNA. And then finally, all of the amino acids are chained together, folded up into their appropriate shape, and the protein is finished. Yay. So now I'd like you to take a second, pause the video, and see if you can walk through that process with the candy factory analogy. how are proteins made? And you want to talk about the nucleus, the DNA, the mRNA, the tRNA, the ribosome, and the amino acids. So see if you can talk through it to yourself, or at least in your head. What is the process of protein synthesis? Now we're going to look at those steps in more detail. The first part of the process, where the DNA gives its instructions to the mRNA, is called transcription. So that is the process of DNA being copied as mRNA. The way this is going to work, and you'll see it on the next slide as well, is that the DNA molecule, so here and here are the sides of the DNA molecule, is going to open up just in one little section, the section that has the gene we're trying to copy. It's going to unzip, and then a complementary copy is going to be made of mRNA. So where the DNA had a C, the mRNA will have a G. And where the DNA had a T, the mRNA will have an A. And now remember, mRNA, any RNA, does not have thymine. Instead, it has uracil. So where the DNA had an A, the mRNA will have a U. It's another way to look at that process. So the DNA, of course, has two strands. Only one of those strands actually gets copied. That strand is called the sense strand. The non-copied strand is called the antisense strand. So here we see the DNA unwinding and opening up. The DNA is the blue, wherever that gene is. Then this orange is the mRNA, which is being copied complementary to the sense strand. And then after that, the mRNA is going to detach and leave. The DNA comes back together. And now the mRNA is going to go through another process, but we will talk about that process on next video. And after that, then the mRNA completely leaves the nucleus, so it just kind of slips out through a pore in the nucleus, out into the cytoplasm of the cell. Now the second part of this process is called translation. So translation is when the Oompa Loompas come with all of the ingredients and put together the candy, or when the tRNAs come with all the amino acids and put together the protein. So again, there's two parts to the process of protein synthesis: transcription, the DNA gets copied onto mRNA, and translation, the mRNA gets translated into a protein. So here is the mRNA. It's left the nucleus and come out into the cytoplasm, and then a ribosome, this gray, sort of clamps onto it, sort of clamps around it. Now the details of this process we're going to look at a little more in the next video, but here's our tRNAs, and they are bringing these amino acids over according to the instructions on the mRNA, and they're being chained together to make this protein. So now, how do we read the code, or how does the ribosome read the code? It can't be just like a one-to-one substitution where like A is going to code for the amino acid serine, because there are 22 amino acids that need to be coded for, and there's only 4 bases. So a one-to-one code isn't going to work. A 2-base code also is not going to have enough. So this system uses a 3-base system. So a set of 3 bases, which codes for one particular amino acid, is called a codon. So for example, the codon GCA on an mRNA means a certain amino acid. One specific amino acid corresponds to that combination. And so every 3-base combination corresponds to one particular amino acid. Now there is some overlap. Some amino acids have more than one codon that code for themselves, because if we use sets of 3 bases, we end up with sort of too many possibilities for the 22 amino acids, but 1 or 2 isn't enough. So there is some redundancy and some overlap there. I'm going to show you the chart on the next slide. So here's the chart that tells you what each codon, what amino acid each one codes for. So if we just look, say, up in the upper left here, UUU. If you have a codon UUU on an mRNA, the amino acid that is going to correspond to that is PHE, which is phenylalanine. I have a document, a PDF, with this amino acid chart with the full names of the amino acids written out in Schoology, so you'll probably want to look at that one for when you're doing activities, but this was the one that I have on this slide, so we can just use the abbreviations. You'll also notice that UUC also codes for phenylalanine, so we do have some overlap there. This is actually also kind of a fail-safe system. It means if you have a mutation, it won't necessarily ruin the entire protein. If you look at this box, for example, UCU is serine. If there's a mutation and you end up with UCC, you still get serine. Or UCA, you still get serine. So in some cases, you can have a mutation that's actually not going to damage the protein, particularly if it's that last base of the codon that gets substituted. All right, so I'm going to quiz you, see if you can figure out which amino acid I'm thinking of before I say it. So how about CUG? CUG, which amino acid does that codon code for? Three, two, one. Correct answer is leucine. CUG is leucine. Let's try one more. How about AGG? AGG. Two, one. Arginine. AGG codes for arginine. Another thing about this code is that this same code applies to every living thing. So essentially, bacteria speak the same language as you, speak the same language as plants, speak the same language as fish, whatever it is. Your cells all speak this same language of DNA. AGG means arginine in you, and it also means arginine in a bacteria. That's why we can actually take a gene from you, like a gene for insulin, stick it into a bacteria, and that bacteria can read that gene just fine, and it will make human insulin, which is how we make insulin for diabetic people. Now, you might have noticed that there are some kind of special codons in here. There are 3 codons that are stop. So those 3 codons do not code for an amino acid. What it means is, as the ribosome is reading the mRNA, if it gets to one of those 3 codons, the ribosome detaches and falls off. So that's the end of the protein. So you're always going to have one of those codons at the end of a protein. There's also a codon that you'll always have at the beginning of a protein, and that is this one right here, AUG. Remember that one. AUG is doesn't say it on this, it will on the one I have in the PDF, but that is the start codon. So the ribosome will not start translating until it gets to an AUG codon. AUG also codes for methionine, so an amino acid will be inserted there as well. Here's a practice table for you. Go ahead and copy this into your notes. The next part of the video will show you the answers, but I'd like you to try this on your own first. You can use that PDF with the amino acid chart that's in Schoology. I'll do one of them, like this one since we just talked about it, methionine. The codon for methionine, or START, was AUG. For this column, if the mRNA says AUG, what did the DNA that it was copied from say? Well, the DNA would have said T, where there was the A, where there was the U, that would have paired with an A, and whereas there's a G, that would have paired with a C. Remember that DNA has T and mRNA has U. Any RNA has U. All right, so go ahead and pause the video and give this a try. And here are the answers to that chart. I think it's slightly ironic that UGA, which is a stop codon, the DNA sequence for that is ACT, which is like the opposite of stop. Go ahead and pause this if you need time to check your work. So here is an example of a task that you should expect to need to be able to do potentially for AP exam, or at least for an exam on this unit. Here is a DNA sequence. Your job is to transcribe this DNA into an mRNA sequence and then translate that mRNA sequence into amino acids using the chart. So just to get you started, if this DNA has a 3' end over here, I would have a 5' end of an mRNA. Where there was C, I would have G. Where there's A, I would have U. Where there's T, I would have A, etc. So first try to turn all of that into the mRNA, then the amino acids. And I'll give you a hint. You have to, when you're translating into amino acids, start with AUG. So you're going to scan your mRNA and try to find the first place that you have AUG. That's going to be your first set of 3. And then the next 3, and the next 3, and the next 3. So go ahead and pause the video again and give this a try in your notes. And here is our answer. So here's the mRNA. I broke it up into sets of 3. The first AUG, you would have had GUAUG. There's my first AUG. That's my first set of 3. So then that corresponds to methionine. And then I'll just take my next set of 3, AGC, which corresponds to serine, and so on until you get to that stop codon, which just means stop. So then here is my sequence of amino acids that I should have come out with.